Hi there, my name is Ryan Tickerson. I'm a student in the Brody School of Medicine class of 2025, and this is going to be our scrubs video detailing the formation of the bilaminar and trilaminar disc. As in all of our videos, here's our mission statement for scrubs, really detailing that we are a student-driven initiative that's aiming to develop supplemental resources for current and future cohorts that will pass through Brody. We participate in a variety of subcommittees to work on these resources for students, and they're made by students. And our hope is that this will eventually become a staple of Brody's student body, exemplifying the unique collaborative community that Brody offers. As always, we have a disclaimer with all of our resources, indicating that these resources are made as a supplement to the primary course materials. However, they are not a replacement by the instruction of the Brody faculty. We hope that you utilize these resources as a supplement and that it aids you in your learning. With that, let's go ahead and dive into the material. In order to think about the development of the bilaminar and trilaminar disc, we first need to go back and do a quick review of where we're at in development. So around the end of week 1, we have formed a blastocyst, which is made up of the trophoblast and the embryoblast, which is that inner cell mass, and we have the blastocystic cavity. Remember that the trophoblast has also divided into the cytotrophoblast, which is the more internal structure that is mononuclear cells, and the multinuclear cells, which is the syncytiotrophoblast. Remember, the syncytiotrophoblast is that component that is going to invade into the endometrium of the uterus and is going to be producing hormones such as beta-HCG, which is going to promote the pregnancy. Right. So in this video, we're going to talk about how we go from this blastocyst into the bilaminar and trilaminar disc, and that's going to take us through the second and third week of development. Okay, and when we're thinking about this, this is primarily going to be changes with the embryoblast, or that inner cell mass that we're talking about when we're talking about the second and third week developments, although there will be a little bit about the placenta later in the video, which is primarily composed of trophoblast cell layers. Okay, so we started with the blastocyst, which we just saw. Now we need to form the bilaminar disc. How this happens is we first form the hypoblast. So the inner cell mass is going to have a layer that is going to form cuboidal cells that is going to be directly in association with the blastocystic cavity. Okay, so this is going to be called the hypoblast. Right above that, we have a layer called the epiblast that is forming. Now, while we're in the uterus, there is going to be an additional amount of fluid that is going to fill the space between the epiblast and the cytotrophoblast, which is right up here. And that is going to be called the amniotic cavity. Because the amniotic cavity is lined by amnioblasts, which are just basically components from the epiblast that have surrounded the space. So this is the amniotic cavity. So now we've formed two layers. We have the epiblast and the hypoblast. The hypoblast are directly beside the blastocystic cavity. The epiblasts are directly beside the amniotic cavity. Remember, we still have our syncytiotrophoblast up here, which are multinucleated cells, and we have our cytotrophoblast, which is surrounding our entire structure. So now that we've formed the hypoblast and epiblast, the hypoblast cells are actually going to migrate. So we're going to migrate from down here in this region, directly in association with the blastocystic cavity, and they're going to surround this entire blastocystic cavity. So when they're surrounding this cavity, they're going to form a membrane. We're going to call this membrane the exocoelemic membrane. So the hypoblast cells are migrating, forming the exocoelemic membrane, which we can see here in the second image. And that is then going to form the space that it's going to change names from the blastocystic cavity to the primary umbilical vesicle. Okay, this can also be referred to as the exocoelemic cavity, but from here on out, we're going to refer to it as the primary umbilical vesicle. Okay, so this is surrounded by hypoblastic cells, which formed the exocoelemic membrane. And again, we have our two cell layers, the epiblast and the hypoblast. At the end of this is around day 9 of development, we have the primary umbilical vesicle and the amniotic vesicle. All right, so this is the formation of our bilaminar disc. Now, we're going to continue to develop, and we need to form the extraembryonic coelom. So how this happens is we start to produce mesoderm, and specifically extraembryonic mesoderm. So remember this term, because we're going to have intraembryonic mesoderm, we're going to introduce later. This is extraembryonic mesoderm. So this is produced by the hypoplastic cells that lined the extracoelemic cavity. start producing this mesoderm, or connective tissue, and this mesoderm is going to start to expand all the way around the amniotic cavity and the extracoelomic cavity to create a space between the developing embryo and the cytotrophoblast, which are surrounding. So as they're creating this space, they're going to expand, and they're actually going to expand to the point where there's spaces that rise between these the layers of mesoderm. And these layers, or the space that arises, is going to be called the extraembryonic coelom. Right? So the extraembryonic coelom is starting to form, so it's the space that is forming between the mesodermal cells that are being made by the extraembryonic coelom membrane. And when this space forms, we're actually going to have separation of the mesodermal cells into two different layers. So we have mesoderm that is directly surrounding the developing embryo, and we have mesodermal cells that is in association with the trophoblast layers, so the syncytio and cytotrophoblast that are surrounding the entire space. Okay, so what this is going to be called is the layer directly in association with the developing embryo is going to be called the splanchnic mesoderm. Okay, so extraembryonic splanchnic mesoderm, and that is going to surround the developing embryo. The component that is in association with the trophoblast layers is going to be called the extraembryonic. So again, this is all extraembryonic mesoderm, but specifically somatic mesoderm. All right, so our two layers that have formed are the splanchnic mesoderm and the extraembryonic somatic mesoderm. And it's important to note that this connecting stalk, which is going to give rise to the umbilical cord in the fetus, this is going to be formed by the somatic mesoderm. So the extraembryonic somatic mesoderm giving rise to the connecting stalk. Right. So at this point, we have now formed the amniotic cavity. We've got the primitive yolk sac. We started to form the extraembryonic mesoderm, which is then going to continue to grow in space till we form two distinct layers with a big space in between. That is going to be the extraembryonic splanchnic mesoderm and extraembryonic somatic mesoderm. And the space between we're going to call the chorionic cavity. All right, so the chorionic cavity forms to involve this space. So this was the extraembryonic coelom that was initially forming, giving rise to the chorionic cavity. Now, something you might have noticed on the last slide, but we didn't go into detail, I'm going to define here, and it's the formation of the secondary yolk sac. So as we're developing, the hypoblastic cells are going to migrate again, and they're going to go around the primary yolk sac and start to shrink it in size. So when they do this, they're going to pinch off a portion, and this portion you can see down here is going to eventually be referred to the extracoelomic cyst. What we really want to keep track of is as they're pinching off a portion of the primary vesicle, they're going to create a secondary vesicle. So this is going to be the secondary umbilical vesicle. You'll notice that you also see the umbilical vesicles referred to as yolk sacs. So this is our secondary yolk sac. All right, and this was due to the migration of the hypoblastic cells. All right, and as this continues to happen, we have now completely separated our developing embryo from the trophoblastic layers, along with the extraembryonic somatic mesoderm. All right, so once this happens, we're going to get a change in name. So these layers of the trophoblast, the syncytiotrophoblast, the cytotrophoblast, and the somatic embryonic mesoderm are now going to be named the chorion. Okay, so they change names to the chorion. That refers to all of those structures combined. The extraembryonic coelom, as we mentioned previously, it is now going to be changing names to the chorionic cavity. So the chorionic cavity is completely surrounding the embryo, or the developing embryo, except for that connection of the connecting stalk, which, if you recall, was formed by extraembryonic somatic mesoderm. All right, and now we have our amniotic cavity. Our bilaminar disc is still in development, so we have our epiblast layer, our hypoblast layer, and now we have a secondary umbilical vesicle. One thing to point out about the secondary umbilical vesicle that you need to know is that this is going to be the location of primordial germ cells that are going to develop. And primordial germ cells just mean these cells are going to eventually develop into the male and female gonad system that allow for reproduction. All right. So these are the primordial germ cells situated within the secondary umbilical vesicle. All right, so this brings us to the end of the second week of development, and a lot has happened. So we're going to go through kind of a summary just to make sure we're on the same page. So we started with the blastocyst. Remember, the blastocyst had trophoblastic layers, and there were two layers specifically. There's a cytotrophoblast, which is the more internal layer, and a syncytiotrophoblast, which is the invading layer that is producing hormones, specifically beta-HCG. All right, we also had the inner cell mass, which was known as the embryoblast, and we had the blastocystic cavity. Along the way to the second week of development, we developed the hypoblastic layer and the epiblastic layer. We also developed the extracoelom cavity, which was known as the primary umbilical or uterine vesicle. This is eventually going to degrade into the secondary umbilical vesicle. We also started to form an amniotic cavity. All right. Additionally, in this process, we formed two layers of extraembryonic mesoderm. There was a splanchnic layer and a somatic layer. The splanchnic layer is in direct association with the developing embryo. The somatic layer is in association with the trophoblastic layers, which is then going to be referred to as the chorion, with those three layers being referred to at the same time. The extraembryonic coelom, which was forming between the two layers of mesoderm, is eventually going to change names into the chorionic cavity, as is surrounding the developing embryo, except for that one location, the connecting stalk, which is going to give rise eventually to our umbilical cord. And remember that the connecting stalk is being formed by the extraembryonic somatic mesoderm. Right, and that's going to bring us to the end of the second week of development. So our bilaminar disc is formed, and this is our current state at day 13 or 14 of development. So the next step is that we have to undergo transition from the bilaminar disc to the trilaminar disc. And this process is referred to as gastrulation. So the way this forms is the epiblast layer is going to start to have a streak down the middle of thickened epiblastic cells. And this is just pretty much a straight line that's forming in that epiblast layer. And as it proceeds, it's going to start in the caudal region, which refers to the tail of the human, and it's going to proceed cranially towards the head. All right, near the head, or the eventual cranial region, there's going to be the formation of what we call a primitive node. Okay, and this is going to be at the cranial head. So once this forms, we have the primitive node. We can now tell where the rear of the developing embryo is and where the head of the developing embryo is. So we've now established an axis for caudal and cranial. Okay, and this is all happening in the epiblastic layer. Now, in between, or around that streak, we're going to start to develop a groove. And this groove is pretty much an invagination of tissue that's diving towards the hypoblast layer, which is directly beneath. And this is what we're talking about when we talk about gastrulation. So the epiblastic cells are going to start invading down underneath the epiblast, and some of those cells are going to replace that hypoblastic layer. All right, these cells that replace the hypoblastic layer are going to be referred to as embryonic endoderm. So that's the first layer of cells that invades. The second layer of cells that invades are going to be directly over top of the endoderm. These are going to be the intraembryonic mesoderm. And remember, this is the intraembryonic mesoderm, which is different from the extraembryonic mesoderm we talked about in the development of the bilaminar disc and the associated structures. All right, this top layer of epiblast is now going to be referred to as embryonic ectoderm. So at this point, we have now had the streak form around which we formed the primitive groove. The epiblast are diving into the groove, and they're going to deposit in three layers. The bottom layer is going to be the endoderm. The second layer is the intraembryonic mesoderm, and the last layer is the ectoderm. Okay, so these are going to form the three layers of our trilaminar disc. And now we have an axis to define anterior, posterior, and cranial and caudal. So remember that the primitive pit is going to be the more cranial portion of the embryo. And down here, this would be the caudal portion. So we have cranial to caudal. And then additionally, we have an anterior and posterior differentiation. So the ectoderm layer which is going to be on top here, that's actually going to be the posterior aspect of the developing embryo, whereas the endoderm layer is going to be the more anterior aspect of the developing embryo. So now we have our axis for orientation. And really, up until the point that you have the primitive pit forming, it's difficult to say what the correct axis is, but at this point, we can now start to define things as cranial, caudal, posterior, and anterior. So the trilaminar disc is really important to understand because it's forming 3 germ layers that are going to give rise to all the structures in the human body. So the 3 layers that are forming are the ectoderm, which is primarily going to be external structures such as the skin, the hair, the nails, sweat glands, as well as giving rise to the nervous system. And we'll talk later in another video about how this occurs, because understanding how ectoderm gives rise to neuroectoderm, forming the nervous system, is a really important concept that really demands its own video. The intraembryonic mesoderm will eventually differentiate into different components, which you'll learn later in the course, but think about the mesoderm as giving rise to all the things that help you to move. So you have your muscles, you have your bones and cartilage, you have your reproductive organs, your blood vessels, heart, and your kidneys. There's also going to be some additional structures, but we'll talk about those a little bit later. The endoderm, think about this as the lining of your GI system and the lining of all your glands. So you have the lining of the digestive system, you have your liver, your thyroid gland, your pancreas, and the lining of the respiratory tract. And again, this is just a very general overview, but it's important to understand that these 3 layers of the trilaminar disc are going to eventually give rise to all of the structures that are in the human body, so we can always backtrack a structure that is in the adult to one of these 3 layers. And eventually that's something that you're going to want to get pretty familiar with as you move forward throughout the course. Now, last thing about development in this stage, during the 3rd week, we're getting continued development of the placenta. And remember, the placenta is really being derived from the layers of the trophoblast. But we're going to talk about the 3 different steps. So you have primary villi formation. And so the primary villi is finger-like projections that are forming within the chorion, invading into the endometrium. Remember that the chorion is specifically referring to the syncytiotrophoblast, the cytotrophoblast, and as well as the somatic, extraembryonic somatic mesoderm. All right, so when those 3 layers start to invade, forming finger-like projections, that is a primary villi. A secondary villi forms when you really have that somatic mesoderm projecting as well. So let me clarify the difference between the primary villi and secondary villi. The primary villi does not have the somatic mesoderm. The secondary villi has the somatic mesoderm invading as well. Okay, so that's the difference between the 2. Now, mesoderm, which we just mentioned, it is eventually going to give rise to all the blood vessels, vascular supply as well. So now that you have somatic mesoderm invading, eventually blood vessels are going to begin to form. And when those blood vessels begin to form, we're going to call that the tertiary villi. So we go from primary to secondary to tertiary. The big differences being primary just contains the syncytiotrophoblast and cytotrophoblast layers. Secondary adds in the extraembryonic somatic mesoderm. And then tertiary is going to add in the blood vessels. And once all of this forms, you have your tertiary villi, you are now going to have utero-placental circulation, which is just a fancy word of saying that blood is coming from the uterus or mom to the placenta, fetal side, and this is allowing circulation between maternal blood flow and fetal blood flow. Okay, I know that's a lot of information, but with that understanding of bilaminar and trilaminar disc formation, we're now going to talk very briefly about some clinical anatomy that's relevant to this topic. The first clinical correlation I want to talk about is sacrococcygeal teratomas. And to talk about this, we're just going to first define what a teratoma is. So a teratoma is any germ cell tumor that contains a variety of tissue types. And this can be, it means it's going to contain ectoderm, mesoderm, or endoderm, or all 3. All right, so it usually contains 2 or more of these germ cell layers, right? And in sacrococcygeal teratomas, so that's down here at the sacrum, or the tailbone, this is going to cause because of persistence of the primitive streak in that caudal pole, so it doesn't completely close down, so you still have those 3 cell layers later on in development. So this is abnormal, but again, this teratoma, which is going to be at the sacral region, is going to have multiple germ cell layers, which is how you can help to identify it as a teratoma as to any other kind of tumor. And the second and last clinical correlation I want to bring up here is a very brief overview of a hydatidiform mole. And so the hydatidiform moles, they kind of present as a cluster of grape appearance, is how you usually see them. But this is under the category of gestational trophoblastic disease. And so what I want to point out with a trophoblastic disease, remember this is our syncytiotrophoblast and cytotrophoblast. The syncytiotrophoblast is producing beta-HCG. So if you have abnormal growth of the trophoblast, you're going to have additional beta-HCG production. And this additional beta-HCG production is going to cause a lot of the symptoms. The excessive nausea and vomiting, as well as the hypertension and the uterine enlargement comes from uncontrolled growth of this molar pregnancy. You're also going to have absence of fetal heart sounds because you have the trophoblast that's replicating, but you don't have the actual development of fetal structures. The second thing I want to point out, and again, just very general, is that there are two main categories for molar pregnancies. You have a complete mole and a partial mole. A complete mole is caused by fertilization of an empty egg and two sperm. So there's no nucleus within the egg, and then the two sperm fertilize it. That means that you're not getting any components from the maternal nucleus. That means that you do not have normal fetal tissue within this pregnancy. This is also going to confer a high risk of choriocarcinoma, which is a cancer of the trophoblastic structures. All right, and this is a very pervasive cancer and spreads quickly. A partial molar pregnancy is instead of being an empty egg being fertilized by two sperm, it is fertilization of a normal egg by two sperm. So this means that you're going to have 3 sets of genetic material. Now, because you do have maternal and paternal aspects to this molar pregnancy, you can get some normal fetal tissue, but you're still going to primarily have the trophoblastic disease, where it's spreading, producing beta-HCG, and it's growing very rapidly. Now, this is less likely to be malignant in the future, but it still is not going to result in a pregnancy that will confer to completion. All right, so again, hydatidiform moles, they're a component of trophoblastic disease, meaning that you're going to have high levels of beta-HCG production and all of its associated effects, and it's going to present as a cluster of grapes. The two most common kinds are complete and partial moles. Complete moles being formed by an empty egg fertilized by two sperm, a partial mole being formed by a normal egg that is fertilized by two sperm. Both of these are going to result in inadequate pregnancies. All right, and that is going to bring us to an end of this video.